it. You know, we all have our needs and desires.
Yes.
But when you substantially increase the lifespan using the force of natural selection, you produce an animal that is actually pretty spectacular. It doesn't just live longer. It's hugely resistant to stresses. It's athletically much more accomplished. Has a much greater total reproductive lifespan. It's a superior animal. So you might ask, if you have a sort of layman's understanding of evolution, well, gee, why doesn't evolution do that for everything? You know, why aren't we all as fantastic as we could be?
That's right.
And there's several. Yeah, of course.
Why not?
There's several answers. Why not? Well, you know the famous scene at the end of Gone with the Wind where Red Butler says to Scarlett O'Hara,
yes.
"Frankly, Scarlett, I don't give a damn."
Yeah.
That's natural selection to us when we're older. That's what the declining force of natural selection means. It means, frankly, when we get older, natural selection doesn't give a damn.
Okay.
That's the sad truth.
That's the sad truth.
But that's the sad truth. In the lab, we can make natural selection care about the survival of any organism whose reproduction we can control, whose life cycle we can control. And we can make evolution answer the question, gee, evolution, how can I build a longer-lived fruit fly? We can also ask, gee, evolution, how can I build a much longer-lived mammal? And that was the proposal I made in 1984, you know, now, what, 25 years ago.
Oh, yes, a long time.
25 years ago, long time ago. I don't know if you were alive then, Justin.
I was, believe it or not.
Oh, very good. I proposed that we use the force of natural selection to build a longer-lived mammal. It was, you know, the first Methuselah Mouse proposal. And that we then reverse engineer that longer-lived properties from the mouse to humans.
Right.
I didn't make that proposal thinking it would be the complete cure. I just thought of it as something that we could make a lot of progress with. And I spent years, basically from 1984 to the year 1999, trying to convince people to take this project on. And as you know, due to my lack of success, I failed. And but in 1999, I realized that the new genomic revolution of the last 10 years gave us the opportunity to go from longer-lived fruit flies genomically to figuring out the controls on aging in mammals to a partial extent.
Whatever overlap there would be between, or analogous genes, you're saying, from fruit flies to mammals?
Exactly. And the technical term we use for that analogous is actually orthologous.
Orthologous.
What it means, orthologous,
okay,
is genes that we share with fruit flies that are recognizably the same in genomic tests. If you'd asked me in 1984 how many of those are likely to exist, I would have said, you know, a few hundred, maybe just a few % of the human genome.
right.
So in 1984, I thought that the idea of going from longer-lived fruit flies to human aging was hopeless. And that's why I proposed the mouse project. And I was wrong, I'll say, in several respects when I made that assumption. It turns out, from genomic research of the last 10 years, that by different estimates, something like 50 to 75% of fruit fly genes are in us.
Wow.
Which is an amazing fact.
That is
I
a
never
lot.
would have believed it's 20. Yeah, a lot. We are something like 50 to 75% fly. Yeah. You may have heard of the movie
Yes,
The Fly.
The Fly, yes.
First version is with Vincent Price, and the latter-day version with Jeff Goldblum and Gina Davis.
Right.
And bad things happen to him because he goes through a transporter and gets fly cells scrambled up with his human cells, and he starts to become a fly-human cross.
Right.
Well, ironically, for that movie, we're already, you know, pretty heavily fly. You know, which in 70s slang is a very cool thing to be, but from the standpoint of the prospect of going from model organisms to humans, it's actually pretty spectacular. And many of my colleagues in molecular genetics take advantage of this fact in going from results with nematodes and fruit flies to humans, and a lot of the excitement about things like the DAF genes, the FOXO genes, resveratrol,
Yep.
is the translation of results from these very simple animal models to medical application. This is actually a huge growth area.
Sure.
In biomedical research.
Okay.
And
okay.
Yeah.
Complete your sentence there. What you were going to say?
As I say, I'm just using those relatively now commonplace ideas, adding the evolutionary biology of aging to it, and coming up with a plan for substantially postponing human aging.
Okay, which makes me wonder, when you first came onto the scene, anyway, when I first became aware more in depth of your work and with the fruit flies, the biologically immortal fruit flies, the question was, of course, how are we going to do this? How are we going to figure out what we can do to retard or reverse human aging if we're just using the fruit flies? Now, many people said, well, we're going to have to breed mice. That's the next step. We're going to have to go to mammals, and it's going to take forever to breed an extremely long-lived mouse, and then figure out what genes are in that mouse and translate them to human genes or our orthologous human genes. And that was kind of a criticism, saying, hey, why don't we just fix the damage that's occurring within our body? We can find some targets and hopefully that'll improve our health, and it's a much simpler method than breeding many years of mice, many generations of mice. But now it appears to me what you just said is that perhaps we don't even need to do that because of the genomics revolution now, that we can read so much more of the genome that we can see what's in fruit flies, what the orthologous genes are in humans, and perhaps we can just use flies and we can find quite a few things that will help retard aging. Is that what you're saying right now?
Yes, you've done a much better summary than I've managed.
Oh.
I'll say that the flies are a first step, and the second step would indeed be a mouse selection program. And to the extent to which we have success with flies, we'll have vastly more success with mice. So I'm saying, first we'll start small, literally, then we'll get a little bit bigger, and as the genomic revolution proceeds, we'll get better and better at doing these things, and I'm very confident that this strategy will be employed in this century, and you will see absolutely radical life extension coming from it.
Okay, and that just brings up, someone asked a question ahead of time before the program I wanted to get to, which we're kind of on the topic right now. Are there any other benefits to working with flies besides their short lifespan and their larger sample size that you can achieve?
Well, the huge benefit is that we know more about fly genomes than almost any other animal, including humans.
Okay.
You basically only want to do this project with an animal that has tremendous genomics. We know all the genes in the fruit fly, and we know a lot about what those genes do. It's what's called heavily annotated. We know more about fly genes, really, I think, than any other complicated animal. The main competition comes from the nematode. And, nematode and fly people are always going back and forth, frankly, being kind of bitchy about it. But
Sure.
There's no question that the fly Drosophila is a spectacular starting point for a biological research program, even one that's really focused on concrete medical applications.
Okay. And since I'm getting into some of the questions that people had asked ahead of time, here's one that's a little bit longer. I hope you can follow, and I hope I'm able to translate it to you in good enough manner here. Given that the method of generating, and perhaps we've already kind of touched on this already, but given that the method of generating long-lived flies in the lab is by selection for longevity traits, which is in effect a form of directed evolution, have you considered that there is selection in the wild for a particular lifespan that is optimal to the survival of the species, and therefore may be interpreted as a negative selector against longer lifespan? If so, could the lifespan be related to the frequency of each generation, which in turn is related to the evolutionary rate, i.e., a longer generational span would imply, all other things being equal, a decreased rate of evolution and therefore a greater risk of failure to adapt to changing environmental conditions.